All right, everybody. Welcome to Chapter 9B. This is the back end of the chapter on muscles and muscle tissue. And what we're looking at here is the, the action of the muscle on a broader scale. Okay. Um, Contraction of the muscle fibers we learned about in Chapter 9A is brought about by the cross-bridging cycle, where myosin engages actin and in the presence of calcium and ATP can bind, pull, release, bind, pull, and release over and over again until we run out of either ATP or calcium. Okay? When this happens at the muscle fiber level, okay, it generates tension, which is the force exerted on the load that's going to be moved or that you're going to attempt to move, right? And so these these contractions that go on at the muscle level can be termed either isometric or isotonic. In an isometric contraction, the muscle exerts force and generates tension, but the dimensions of the muscle don't change. And so that would be something like locking your hands together in front of your chest and then pulling, okay? And you're generating tension and you're burning energy and you're actually building muscle strength when you do that, but your muscle dimensions aren't changing at all. Okay, that's why it's called isometric. Iso means the same as, right? So the same metric, meaning the same length of the muscle throughout the contraction. Isotonic is where the muscle either increases or decreases in length while it's producing tension. Okay, um, if the muscle is decreasing in length, that means that the force the muscle is generating is greater than the load. And if it's increasing in length, it means that the force produced by the muscle is less than the force generated by the load. Okay? So the way that we pull this off at the muscle level is that we can recruit more muscle fibers into the activity, or we can generate more force out of a single muscle fiber. Okay? And there's a few tricks that allow us to do that. So remember that each muscle is served by at least one motor nerve, which contains lots of motor neurons. Okay, remember that peripheral nerves are basically bundles of axons, and in this case, they would be myelinated axons, and they branch out at the axon terminal into um, many forks that have those little synaptic boutons, those little swellings at the end that are filled with neurotransmitter, and they come into contact with many, many different targets. So a motor unit is defined as a single motor axon, okay, a single um, a single extension from a single nerve cell, and all of the muscle fibers that it talks to, okay, and you can see here sort of how that looks. The red and the purple represent two different motor units, and the muscle fibers you can see in the cutaway. All right, and one of the ways that we can produce more force is to bring more motor units into the activity, right? The more fibers that are engaging in the activity, the more force you're going to produce because the more cross bridging is going to go on, right? And that means you're going to be able to move a bigger load. One of the things that happens when you are trying to exert force against a load is that these motor units will switch off automatically in order to prevent fatigue. So motor unit 1 might fire for a bit, then it'll stop, and motor unit 2 will fire for a bit, then it'll stop. And that's just a way to try and prevent you running out of ATP real quick, okay, while maintaining the activity, all right? Bottom line, though, is the more motor units are playing, the more force you're going to produce, right? One of the things that we can do in order to examine a contraction is to do this in a lab setting, right? And the way we used to do this was to use an actual frog gastrocnemius that we would dissect off of a recently killed frog, and we would attach it It's probably easier to draw here. Let's draw the setup. Okay. So we would do, let's do a pen. Okay. So it would look like this. You would have a bar, all right, and then you would have a projection out here, and then the frog gastrocnemius would be here. Right, this is the muscle. Okay, we'll color it in red. Right, and then there would be it would be extended from this, right, and then at the other end there would be a clip that would extend it here, and then there would be a bar that comes out, and it would be basically a structure that would be moved. It would have a pin at the end, all right, and then there would be, and this would be just the base of the assembly, and then let's switch colors. There would be a rotating drum out here, all right, that had chart paper around it, and basically it would spin. So I'll draw the direction of spin here, right? It's spinning like this, okay? And as it spins, we would stimulate the frog gastrocnemius and it would contract, and then the pen would go up and down on the chart paper and draw on it, okay? And that would produce something called a myogram, and that would be a basic recording of the activity that we can produce artificially in a muscle, right? So we don't do that anymore. We use devices that either mimic this kind of experimental setup, or we can do experiments where we pass current through a living organism without dissecting it. Okay, so the three phases of muscle twitch that you can record on a myogram are the latent period, where we get ready for the excitation contraction coupling, the contraction period where the muscle's actually shortening, and the period of relaxation where the calcium is going back into the sarcoplasmic reticulum, the cross bridging is ending, and the muscle fiber goes back to its resting length. Okay. Note that a muscle fiber is going to contract more quickly than it's going to relax. That's because not all the fibers are going to relax at the same time. Okay. So if you generate a simple stimulus, okay, like here in dark blue, where the red arrow is the stimulus, and what will happen is that you'll produce a twitch, okay, and this is not something that muscles normally do in the body. You don't normally go around performing actions by twitches, okay. This is something that's really um, something that you see in a lab experimental setting, okay. But what will happen here is you can generate a stimulus like an electric current through a muscle, right, that you've dissected out, and then you'll have the latent period, the contraction period, where we're generating the tension, and then the muscle will slowly relax, okay, and that is what we call a simple twitch. All right, different muscles will twitch at different rates, okay. So you can see here some different muscles from different locations in the body. The extraocular muscle is the one that makes you here look left with the left eye or right with the right eye, right? The gastrocnemius is the muscle that runs across the back of your knee and the back of your ankle and lets you bend your knee and point your toe. And soleus runs across the back of the ankle. And you can see here that the same stimulus applied to three different muscles results in twitches of different lengths. And the reason for this is that the muscle fibers that make up these different muscles have different metabolic properties. Okay, some are designed for generating tremendous power but are easily fatigued. Some are designed for long-term work and don't fatigue very easily, and then some are in between those two types. Okay, so this is again a lab phenomenon. When we move in the body, what we do is we sort of take the simple twitch and we use that as a component of what generates contractions that generate movement in the real world. Okay, so you generate these graded responses that are going to be appropriate for the force they're trying to apply, right? This is important for movement and for posture. And we change the response by either changing the frequency of the stimulation or the strength of the stimulation. Okay, by frequency of stimulation, we mean the number of action potentials per minute. Okay, and by strength of stimulation, we mean the number of neurons that are actually firing at the same time and talking to the different motor units. Okay, so you can see here in a single stimulus, we generate a contraction, relaxation, we get up to maximum tension for that muscle fiber in a single twitch, right? And then we go back to the state we were prior to the stimulus, right? So this is the latent period, right? This is the period of contraction, period of relaxation, okay? So what happens in the real world, right, is that we stack those simple twitches, one on top of the other, okay? And what happens here is by increasing the frequency of action potentials to a muscle target, we generate more power with each successive stimulus. And the reason for this is that the calcium inside the muscle doesn't get a chance to get pumped completely back into the smooth endoplasmic reticulum, and so it's easier for the myofibrils inside the muscle fiber to re-engage in cross-bridging and produce more power. And you generate a process called TREPI, where we get this stair-step effect up to what we call unfused tetanus, right, or incomplete tetanus, where we're generating the maximum amount of force while still having a period of relaxation in between these muscle contractions, okay? So that is one of the ways that we can produce more power within a single muscle fiber, right? We generate more action potentials, we get them come and run one right after the other, and the fibers generate more force for us, okay? If we keep on increasing the frequency of the stimuli, we will eventually get to something called fused tetanus, where the muscle is in a state of contraction, the muscle fiber is in a state of contraction, and it does not relax. Okay, there's no relaxation between the stimuli. What happens here is you very rapidly run out of ATP, okay, because the rate at which you're consuming energy is far greater than the rate at which energy is being delivered to the muscle. And so you eventually go into fatigue. So that is illustrated here, right? See the rapid volley of stimuli. We're in fused tetanus, and then pretty quickly we drop off being able to respond because we're out of gas, essentially. Okay? So fused tetanus is another rare phenomenon. Okay? This would be something that you might see in a life or death situation, like you're holding onto a tree branch over a cliff to save your life. Okay? This is not something that usually occurs. Okay? Generally, when you're engaging in normal movement, what you're doing is some combination of TREPI or unfused tetanus. Okay. The other way that we can produce more power is through recruitment, where we get more motor units to participate in the activity. That generates more power because we got more motor units acting at the same time, and that allows us to move a greater load. Okay. A subthreshold stimulus will not produce an observable contraction, okay, because the number of action potentials is insufficient to cause the muscle cell membrane to produce an action potential that goes all the way down the length of the muscle fiber and into those T-tubules, right, because we don't open enough of those sodium gates at the neuromuscular junction. A threshold stimulus will produce the first observable muscle contraction, and then a maximal stimulus will generate the strongest muscle contraction. Okay, so what you're seeing here at the muscle level, right? The stimulus strength, the first observable contraction is going to be at threshold stimulus, right? And you can see here the number of motor units participating is relatively low. Then, as the stimulus strength increases, right, more neurons are firing, right? We call in more motor units, and the result is that we generate greater power, right? Up to what? Where all the motor units are in play, right? And that would be at maximal stimulus. Anything above maximal stimulus isn't going to generate more motor units because they're already all participating. So you can see here with the strength of contraction, we build and build and build until we get to a peak, and then even though the the strength of the stimulus is greater, right? We're not getting any more motor units in the activity, and so we don't generate any extra force. Okay, so two ways that we can increase the participation or the force generated by a muscle. One, by increasing the frequency of action potentials on a muscle fiber, right? And the other is by having more motor units participate. Recruitment normally works on what we call the size principle, where the motor units that talk to the tiniest muscle fibers come first because the load is minimal. Then, as we have to increase the force exerted against a bigger load, we call in the intermediate size muscle fibers. And then when we got to go full bore, we call in the biggest muscle fibers, right? So you can see here the different motor units and when they come in, right? As the load that we're working against increases, or as the force we want to produce gets greater, we have to call on motor units to talk to bigger and bigger fibers. The different types of contractions are shown here, right? In an isotonic concentric contraction, what happens is that the muscle length decreases and the tension increases. Okay, so you can see here the tension is going up and the resting length is going down, which means that the force exerted by the muscle is greater than the force exerted by the load, and so the load moves. Okay, in an isometric contraction, the resistance produced by the load and the force generated by the muscle are the same. So the muscle doesn't change its shape at all, but it does develop tension and it burns energy, okay? And it is a way to do exercise, all right? But nothing is moving, all right? That's what isometric is defined as. See how the resting length does not change here, okay? Now, the other type of contraction, if we go back one, is called an eccentric contraction. And in an eccentric contraction, what happens is that the muscle actually gets longer, all right, because the load is producing a greater force than the muscle can produce. But the muscle is still trying to generate tension. So it would be like you trying to lift an object that's too heavy, right? Like a lead brick off a table, right? You slide it off the table and you're not expecting it to be as heavy as it is, and you try to lift it up, but you can't, right? And maybe you drop it, okay? All three cases burn energy, qualify as exercise, right? And produce tension. Now, in fact, this idea of isometric exercise is something that's very old, okay? Back in the 40s and 50s, there was an exercise program that used to be sold out of the back of comic books by a gentleman named Charles Atlas. Okay, and Charles Atlas wanted to build up his body, and he didn't have access to a lot of exercise equipment, and so he used to go to the circus and look at the big cats in the cages, and he noticed that even though they were in a small space, they still had tremendous muscle tone. And he wondered why that was, and as he watched them, what he noticed is that they did something called isometric exercise, okay, where they would You've probably seen this downward facing dog where their back end's up in the air and the front end is down, and then they switch it, right? And they maintain that tension for a time. And that is muscle on muscle, and that still qualifies as exercise. So he generated an entire exercise program based on nothing but isometric exercise, where there's no movement, but the muscle produces tension, and um, the, 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 the bulk of the muscle increases. Okay, examples would be things like clasping your hands in front of your chest and pulling them in opposite directions. Okay, or putting your two palms together in front of your chest and pushing them together as hard as you can. Right, you're generating tension. You're doing exercise, but there is no movement going on, and you don't need any equipment. An example that you're probably more familiar with is planking, where you get down on the ground like you're going to do a push-up, and you just keep your body at a 45-degree angle, and you don't move anything for about a minute, right? And that's meant to develop your core muscles, okay? Muscle tone is basically a spinal reflex that keeps a slightly contracted state in all muscles, even when you're not really consciously doing movement. Okay, what happens here is that there are receptors inside the muscle tissue and in the tendons that respond to stretch. Okay, and what happens when the stretching is too great on the tendon or the stretching is too great on the muscle fiber is that it'll send a signal to the spinal cord to counteract that, right? And so what will happen then is if the muscle gets overstretched, there are um, uh, essentially receptors inside the muscle called spindle fibers that will sense that and will send a signal to the spinal cord to talk to a motor neuron that tells that muscle fiber to contract in order to oppose the stretch, right? And the same thing goes with the tendon, right? If the tendon gets overly stretched, it will, through something called the Golgi tendon organ, send a signal to the spinal cord to cause muscle contraction to relieve the stretch on the tendon. And the idea here is to avoid damage to either the tendon or the muscle so they don't tear, okay? And this goes on without us really having to think about it very much, okay? And it keeps the muscles firm and healthy and ready to respond. This is one of the reasons why when you cut a peripheral nerve to a muscle, right, that the muscle will go into paralysis in the form called flaccid paralysis very quickly, and will, if you don't do anything about it, lose muscle tone very rapidly. Whereas if you damage the pathway from the brain to the nerve fibers that talk to the muscle, you'll go into what's called spastic paralysis, where the muscles will undergo periods of contraction and relaxation, but will still not be under conscious control. Okay, so the reason for the spastic paralysis is that when what we call the upper motor neurons are damaged, you still have a connection between the spinal cord and the muscle, and that is enough to keep this degree of tone somewhat in state, right? But it's not the same really as using the muscle to move, and the muscle will over time atrophy as well. Okay. Now the energy that's used to generate muscle contraction immediately comes from ATP, right? But the ATP inside a single muscle cell is not going to last us very long, right? About 15 seconds worth of power is available from the ATP in the muscle and its immediate recharger, which is something called creatine phosphate, which is basically a molecule that has a high energy phosphate bond that it can transfer to ADP to make ATP. Okay, so you get about 15 seconds of work out of that. All right. Your ATP will be gone in about 4 to 6 seconds. That's stuff that's immediately stored in the muscle, right? The next pathway that you draw on in order to maintain the activity is glycolysis, right? Glycolysis is a fast metabolic pathway that causes a net gain of 2 ATP per molecule of glucose, and is something that the muscle can go into if there's insufficient oxygen to maintain the activity. So in this case, you're going to get about half a minute out of running nothing but glycolysis for the cell. So what we're doing here is we're taking glucose from the blood, we're cracking it in half, we're pulling out enough energy for 2 ATP, and that's not going to get us real far, okay, because eventually the rate of ATP consumption will outstrip the rate of sugar delivery from the blood, and the muscle will go into fatigue and the activity will stop. Okay, so that doesn't get us far enough along to maintain the activity. This is the explanation for why you can't sprint for hundreds of miles. Okay, because the rate at which you deliver the energy is going to be less than the rate at which you consume it. All right, and then the muscles will just give up. Okay. You might ask, well, where's the rest of the energy in the glucose? Well, it's trapped in a product called lactic acid. Lactate builds up in muscle that's in anaerobic metabolism, and it ends up going into the blood and back to the liver, and it hangs out there or is stored as glycogen or fat until the activity stops, and then we can convert it back into pyruvate, send it back to the muscle, and let the activity resume. And that's a pathway known as the Cori cycle. Okay. Glycolysis doesn't require oxygen. We generate 2 pyruvates per glucose molecule and a net gain of 2 ATP. Normally, the pyruvate will enter the mitochondria, and aerobic respiration will strip the rest of the energy out and make it available as ATP. At 70% maximum contractile activity, bulging muscles squeeze blood vessels, oxygen delivery is reduced. Okay, pyruvic acid is converted into lactic acid. That goes into the blood, that goes to the liver, and it gets stored or it gets used by the kidneys and the heart. It gets converted back into pyruvate by the liver once the activity is over. Okay, the anaerobic process yields only 5% as much ATP as aerobic respiration, but produces the ATP about double as quick. Okay, so the way to look at this is that if you don't have oxygen around, you're generating ATP 20-fold less than if you do. Okay, because when you engage in an activity that has you consuming oxygen at the same rate as you're delivering fuel, okay, and burning ATP at the same rate that you're delivering fuel, then you can maintain the activity for a long time. Okay, this is why, again, you can hike or jog a long distance, but you cannot sprint a long distance, right? It's a matter of the rate of consumption of the fuel versus the rate of delivery of the fuel. And so here what happens is that we have all 3 legs of metabolism generating energy for us. We have the glycolytic pathway in the cytoplasm. We have the Krebs cycle in the mitochondrial matrix. And then we have the electron transport chain in the inner mitochondrial membrane along the cristae, right? And that allows us to generate close to 40 ATP per molecule of glucose, right? So the result of that is that we get a lot more energy, even though we're generating it more slowly, and that allows us to maintain the activity for a longer period of time, right? So you can see here in this scenario how quickly we burn through this stuff, right? Aerobic endurance is the length of time the muscle contracts when we're doing aerobic exercise, right? We're engaging in an activity that consumes energy at either a slower or at the same rate than the blood delivers it, right? But the anaerobic threshold is the point at which the metabolism goes to anaerobic, and the end of the exercise is going to come about much more quickly, right? So we look at the example of a sprinter, right? In 6 seconds, her ATP is toast, right? 10 seconds, the creatine phosphate contribution to ATP is burned up. In another half minute, the glycogen inside the muscle, which is the glucose polymer that serves as an energy store, is burned up. And then we end up going into energy exhaustion, right? Because we don't have the ability at this point to generate ATP using the electron transport chain because we don't have enough oxygen there, and the exercise ends, okay? So that's why you can only sprint for a short period of time, right? Or power lift for a short period of time, okay? Because the energy consumption outstrips the rate of energy delivery. Why does muscle fatigue happen? You are unable to contract even though the stimuli are continuing to come because you've got an abnormal distribution of ions across the membrane, which interferes with excitation-contraction coupling. Prolonged exercise can damage the sarcoplasmic reticulum and interfere with calcium regulation and release. You generally don't get to a complete lack of ATP because if you did, you would enter something called a contracture, where you'd be in continuous contraction like you get in a state known as rigor mortis, okay, which we talked about in chapter 9a, okay. So what generally happens is the ATP levels get so low that the muscle just can't function. To return to the resting state, we have to replenish our oxygen reserves, convert the lactate back into pyruvate, send that back to the muscle. That allows us to replenish the glycogen stores in the muscle. The ATP and the creatine phosphate reserves are regenerated, and this requires more oxygen, and that's one of the reasons why after the end of the exercise you're sucking wind, right? And that's to get all this stored energy back up, okay? And again, where are you pulling this energy from ultimately? Your fat stores in your body, right? That's, again, your body's pantry, right? Your store of energy when you're not actively eating. About 40% of the energy released when you are doing exercise is useful for work. The rest is heat. And if those levels get too high, okay, then you could end up having heat stroke. It could end up killing you, right? Dangerous heat levels are prevented because we lose heat from the skin through radiation and through sweating, right? Generally, shivering is the result of muscle contractions to produce heat when we're cold, right? And that is a response via homeostasis using the hypothalamus as the control center to try and get the body temperature back up when it drops too low, right? Because if the body temperature gets too low, then eventually chemical reactions will slow to the point that we can't support life and you'll die, right? Same as we don't want the body temperature to get too high, right? If the body temperature gets too high, then we cause the tools of life, proteins, to denature. Then they can't work properly anymore, and the result is that you die, okay? So the body's always working to keep that body temperature within a range compatible with life, okay? All right, well, that brings this chapter to an end. I'll see you guys in the next podcast, and stay safe.